Learning Objective 8 introduces receptors known as TRP receptors. The first thing to think about when it comes to pain characteristics is the location of the pain, and that because of the way nociceptors innervate the body and the different subtypes of nociceptors that exist, there is a different experience that may be transduced because of the pain location. But in general, we talk about 4 categories of pain by location, and that is first, somatic or cutaneous pain, muscle pain, deep pain, so something like a very deep type of bone sort of pain, and then visceral pain. Of course, all of these are signaled through nociceptors, which we learned previously are very specialized sorts of afferent fibers that transmit and encode noxious stimuli. Now, electrophysiologically, we talked about nociceptors could be a C fiber or an A delta fiber. Now, among C or A delta fiber, there are other types of nociceptor subcategories, and primarily going to be based off the sort of pain or the sort of noxious stimuli that they transmit and then respond to. And the 3 stimuli they respond to are mechanical forces, chemical compounds, and thermal environments. Now, the mechanical forces are essentially a mechanical force, everything from a moderate pressure with a blunt object to specifically tissue damaging stimuli. Chemical modalities of pain characterization are those that where nociceptors respond to chemical compounds. Those could be endogenous or exogenous. Endogenously, those are things like the pro-inflammatory mediators, acids, things like that. And then exogenous compounds, such as a very famous one is something called capsaicin. We'll learn more about that in a little bit. But capsaicin is the pungent ingredient found in chili peppers. And then finally, we have thermal response. Now, thermal response indicates that there are nociceptors that respond to temperature, which is kind of an interesting idea. But they can respond to what we call noxious or an extreme heat or cold. Have you ever touched something and it's so cold that it hurts? Well, that's what we're referring to with noxious cold. Of course, noxious heat would be something that would introduce a burning type of sensation, and that is, of course, painful. And the thermal temperatures can, in fact, directly activate the receptors that are expressed by nociceptors. And we will get into that in a little bit. Now we can put the information about pain characteristics by location and modality together in this slide here. It is important to note that stimuli adequate to activate nociceptors are not the same for all tissues because of the modality that is most commonly associated with the location. So let's think about skin. Skin, it being external to our body, is exposed to the elements. It is, in fact, exposed to all 3 different types of pain modalities: thermal, mechanical, and chemical. And so as a result, the nociceptors that innervate the skin are about equal in terms of responding to these types of noxious stimuli. Joints are most likely to be exposed to mechanical and chemical sorts of pain, chemical pains being things more like inflammatory mediators rather than, a venom or a bee sting. But what that means is the mechanical and chemical sorts of pain that affect joints manifest themselves in fairly distinct ways. Now, muscle pain is going to be primarily mechanical and chemical, again, similar to the joints. And then the visceral pain is primarily, it can be mechanical, but it is more likely to be chemical and possibly thermal. But the mechanical pain that really impacts the viscera is usually something that occurs with distension or distension of the lumen of the internal visceral organs, even something like traction on the viscera or the mesentery. And then even probably more likely than mechanical pain in the viscera is something like chemical pain. So inflammation, acid, other sorts of irritants like that. Now the next slide is going to show you kind of a summary of what all of this means when we put it together. So here's a summary of the different types of pain we just went over. The cutaneous or the skin, deep pain, muscle pain, and visceral pain. First row is essentially giving you examples of those locations, kind of what that means and what could happen there. And then the bottom row really kind of talks about characteristics that you would expect because of what we know regarding the types of nociceptors that innervate them and the sorts of noxious stimuli they're exposed to. So, for example, cutaneous pain with the particularly that mechanical pain, the A delta fibers, the C fibers, you get that combination of fast, that first phase pain, and then that followed by that second phase, slow, dull, aching, throbbing pain. Very common in the skin and the cutaneous structures. Deep is primarily associated with bones, ligaments, things very deep internal to the body, and is usually expressed more dull and achy rather than sharp. However, it can be associated with muscle spasm as well. But the muscles themselves, whether through strain, injury, ischemia, or even something like delayed onset muscle soreness, can be fast and slow pain. And then the visceral. Visceral organs, of course, we're talking about the abdominal cavity and the various visceral organs inside. I guess you could also include the heart and lungs in that category as well. Very poor localization, very sensitive to stretch in particular, as well as chemical compounds, and very much associated with referred pain, which is part of that poor localization process. So a food for thought question at the bottom, if you think about osteopathic manipulative medicine and the different types of manipulative therapies that you learn in your OMM courses, kind of begin to assimilate in your mind, how does OMM help with these different types of pain, depending on the types of locations that you're at. Speaking of referred pain, there are two main reasons why we experience referred pain. The first is in order to localize pain. Remember, pain localization is experienced through those A delta and C fibers, and the C fibers are pretty imprecise when it comes to localization of pain. But the A delta fibers really require continual training, particularly early in life, to develop the ability to precisely interpret the localization or the origin of that pain. And while that is, it does happen sometimes in early childhood, the predominant pain sensing, in particularly in that stage of life, is probably going to be more like somatic or cutaneous, maybe muscle pain. But you don't really get a lot of early life visceral pain enough to really thoroughly train your ability to localize where it is within the abdominal cavity or the thoracic cavity. So the brain does require some experience to learn how to localize pain. And essentially, you just don't get that training often enough, early enough in life to really hone in on where to train it. And so in addition to that, if you put that kind of phenomenon together with the fact that you do have some afferent convergence in the dorsal horn, and as well as a few other phenomena we're not going to get into in this lecture series, but you have something called dichotomizing afferents, where you have essentially peripheral branches of one afferent can innervate different structures and essentially just converges higher up. Because of those convergent phenomenon, it becomes very difficult for you to exactly interpret where is the real origin of this pain and where is the referred pain, which is the true pain. So far, we have previously discussed a lot of the different types of pain and why we experience them, but now we're going to transition a little bit into the physiology and the mechanisms that underpin why we feel the different modalities of pain that we feel. And in order to begin that conversation, we need to learn about a family of receptors called TRP receptors. These are TRP receptors right here, and they stand for transient receptor potential. And these are actually a very big family of receptors that consists of, I mean, I think there are over 30 of them recognized now. There might be over 40 by now, actually. And they're subcategorized into different receptor families, and the kind of fourth letter or whatever letter follows the TRP is to be interpreted as to what family it comes from. Now this is a graphic over here on the right that shows you the breakdown of the families. You don't need to know all of this. This is just to show you how many they are and how they're grouped together. The three that I am going to hold you responsible for knowing are these ones over here on the left, and they are TRPV1, TRPA1, and then TRPM8. Now, before we go into each one of these TRP receptors in a little bit more detail, I will explain that they are ligand gated, so that means you have to have some sort of chemical signal that binds to the receptor. So it has to have a ligand. And then it's non-selective cation channel, which means that upon activation or upon ligand binding and an appropriate transmission of potential change, electrochemical changes, you will have opening of the cation channel, and through it can flow calcium, sodium, or potassium. So it's non-selective. It can allow these three different cations to enter. But most famously and most importantly for our purposes, it is a non-selective calcium cation channel. Now I want to talk about the ligands that activate the different TRP receptors themselves. Now, I mentioned on the previous slide that there are 3 TRP receptors I'm going to hold you accountable for, and they are TRPA1, TRPM8, and TRPV1. This graphic is showing a variety of different TRP receptors, and then above it, some of the things that can activate those TRP receptors. Now, let's start with TRPV1. TRPV1 is very famous for being activated by the endogenous compound found in chili peppers, the pungent chemical capsaicin, which is responsible for the heat that you are familiar with whenever you eat a chili pepper type of vegetable. Over here on the left, you'll see this TRPA1. Now, TRPA1 is also found in foods in the environment. And it's kind of an interesting phenomenon how a lot of the TRP receptors are responsive to things that are existing out in the world in our environment. So TRPA1 is responsible for being activated by the ligand allyl isothiocyanate. Now, this is a compound found in a variety of different things that also transmits a sensation of heat and or spiciness, and that might be either garlic, radishes, or even mustard. It's also responsive to the primary compound in cinnamon aldehyde. All of those are ligands that can activate TRPA1. And as a result, because these are receptors that are expressed by nociceptors, the heat sensation transmitted through these compounds are usually translated as something to be painful because, again, these are receptors expressed by nociceptors. They're noxious compounds, and particularly if activated with a great enough stimulus, it's usually painful to people. Now, keep in mind that pain requires an unpleasant sensory and emotional experience. So those of you who quite thoroughly enjoy the spiciness of capsaicin or allyl isothiocyanate, if it's not unpleasant for you emotionally, then it technically isn't pain. It's simply nociception. Now, TRPM8 is interesting. You may not think that TRPM8 is painful because it's expressed by, or it is responsive to the primary ligand found in mint, and that, if made synthetically, is menthol. Now you may not think, well, that's not painful. Mint isn't painful. Menthol isn't painful. It's actually quite nice. Well, that's because there's a little bit of a dose response here that is to be considered. If you poured 100% straight menthol onto a nociceptor, I promise it would be very painful. So you may not think of it, but in fact is a noxious stimuli. And because it's transmitted by a nociceptor, it goes to the same places in the brain as these other ones, and it can be interpreted as painful. Now, I also wanted to quickly bring up another compound here. You're not held responsible for TRPV3, but TRPV3 is responsive to the primary compound eugenol. Eugenol is found in bay leaf, oregano, and thyme. So if you've ever taken a bite of any of those things straight, or particularly if it's been concentrated, it can be quite spicy in its own way and quite unpleasant and painful. So I just think that that's an interesting phenomenon. Now, the other thing that I want to draw your attention on this slide is this lower bar here, color-coded from blue to red. Now, TRP receptors are also responsive to changes in temperature. Actual changes in temperature itself can activate these receptors even in the absence of a chemical ligand. So that's pretty amazing to think about, a temperature-sensitive receptor. Now, the higher ones, TRPV1, the higher temperatures, particularly able to activate the TRPV receptors, particularly V1, V2, a little bit of V3 as well. But it takes quite a bit of actual heat, and part of that is why the painful sensation associated with straight capsaicin or chili pepper is also conflated with feelings of hotness. And that's because it's the same receptor. This particular receptor, TRPV1, can respond to 50 degrees Celsius temperatures, but it can also respond to capsaicin. And your brain doesn't particularly know why TRPV1 has been activated, just that it has. And so that's why a lot of these, the chili pepper taste and the pain associated with that is usually conflated with heat. Now, there's other physiological mechanisms at play as well, but I think that that's it's a big one and it's very important. On the other end of the spectrum, you have other receptors. TRPA1 is shown on this particular graphic here as being responsive to cool. So let's ignore that for a minute because I'll come back to that. TRPM8. Now this is probably something that's familiar to you. If you think about mint, your first thought when you think about mint or menthol might be that kind of cooling sensation. And in fact, that's exactly why it's used for therapeutic purposes, for that kind of cooling sensation, and then for culinary purposes because of that kind of mint crispiness flavor. And TRPM8 does in fact respond to lower temperature ranges, but it takes a kind of an exposure to it in a wide enough area of the body to really activate TRPM8 and transduce that feeling of painful cold. Now let's go back to TRPA1. TRPA1 is, as I mentioned before, responsive to the ligand allyl isothiocyanate and cinnamaldehyde, some others as well. And those are usually thought to be kind of hot compounds. If you think about cinnamon, radish, those are hot, kind of spicy compounds, but it's hot in a different way from the TRPV1 responsive capsaicin. And it has to do with the fact that TRPA1 simply does not respond to the very high heat temperatures that TRPV1 does. And on this graphic, it shows that it responds to cool temperatures. Now, it is shown that way on this graphic because this graphic is designed for those who study this mechanism, which primarily these studies are conducted in lower species, mice, rats, etc. And TRPA1 in the lower species, not mammals, or sorry, not humans, is in fact responsive to the lower temperatures. So it is that way in other species, but not in humans. All right, the first TRP receptor that we're going to go into some more detail is TRPV1. This is expressed by many different types of C fibers. Now, a C fiber could express more than one TRP receptor, but usually not more than two. And in fact, usually it's only one. They are found on C fibers themselves. And again, the TRP is transient receptor potential. And then the V stands for vanilloid. That means that it's responsive to vanilloid compounds. So that's a chemical structure. If you are familiar with your organic chemistry, that might conjure up an idea of what that looks like in your mind. You don't need to know that, other than for our purposes, the most important ligand that activates TRPV1 is noxious heat and capsaicin. So for capsaicin, I've previously talked about that when it comes to the exogenous pungent compound found in chili peppers. But also, and a very important thing that can also activate TRPV1 is the inflammatory mediator bradykinin. And then, of course, those noxious heat, so very extreme heat over 43 degrees Celsius. So not only does activation by one of the activating substances, one of the ligands, or noxious heat, activation of that in a peripheral sensory nerve fiber, it not only leads to an action potential in that afferent fiber, but it also leads to the release of neuropeptides from that afferent fiber. And I'll get into that a little bit farther on in this series. Now, if you have sustained TRPV1 activation, such as, let's say, the receptor is bathed in inflammatory mediator type of soup, then that can lead to further molecular signaling, which leads to eventual kind of a self-propagating cycle here, a positive feedback loop that leads to vasodilation and then further immune cell recruitment, inflammation, probably more bradykinin, which would then subsequently further activate and kind of continually propagate this molecular signaling cycle where it just simply sits there in its own ligand and continually activates itself. For your purposes as future physicians, TRPV1 is implicated in a wide variety of chronic pain syndromes, particularly those of unknown etiology. Migraine is an important component of TRPV1 signaling. Dental pain, cancer pain, inflammatory pain, neuropathic pain, visceral pain, and osteoarthritis. Now, TRPA1, the A in this one stands for anchor and repeats. It's another chemical structure that you don't need to know, but what you do need to know is the name, the TRPA1, and then what activates it. It is allyl isothiocyanate, which is a compound found in a lot of the kind of non-chili pepper hot foods, if you will. It's found in mustard oil, wasabi, horseradish, radishes as well as garlic. And then also it can be activated by cinnamon, so that kind of non-chili pepper heat, if you will. Now, TRPA1, very importantly, is involved in a number of inflammatory pain states, particularly those that involve the viscera, like in irritable bowel syndrome and idiopathic chronic pancreatitis, and even idiopathic chronic prostatitis. Otherwise, it's also involved in chronic itch, which is a very debilitating syndrome. It has no good pathophysiologic mechanism known yet, but we know that TRPA1 is very heavily involved in that, as well as allergic contact dermatitis. Now, you might hear sometimes about this phenomenon called paradoxical pro-nociceptive effects induced by anesthetics. And whenever you hear about that later on in your pharmacology lectures or later on in your rotations, you'll know that sometimes anesthetics induce pain, even though that's kind of their job is to avoid pain. They can sometimes have a paradoxical effect. And when it does so acting through TRPA1. Now, the last TRP receptor I'm going to go into detail on is TRPM8. I think TRPM8 is a really neat TRP receptor, and that is because it's so important to a lot of our daily lives, and whether or not we are familiar with it or not, we're surrounded by TRPM8 agonists. Now, TRPM8 can be activated by the ligands themselves, camphor or menthol, but it can also be activated by innocuous cooling and noxious cold, so essentially just cool temperatures, which is part of why the cooling agents, camphor or menthol or mint, also are conflated in the brain as having a cool sort of sensation. Now, a lot of these topical products can be used for analgesic properties. Now, that may seem a little bit like a misnomer to you because it's transmitted as because it's transmitted by a nociceptor, the C fibers, and that is very specific to the dose of the ligand found within. But here are some examples of over-the-counter products that contain menthol, which we use to take advantage of for their kind of cooling and relaxing properties. Oragel for instance, also has some other ingredients in it that help ease that numbing effect, but menthol is also found in it as well. Learning Objective 9 introduces some basics related to molecular biology of pain. Now let's look at what nociceptors are out in the skin. We know that the axons of nociceptors are either comprised of those slowly conducting unmyelinated C fibers or those thinly myelinated, a little bit faster, but not as fast as a big motor neuron, A delta fibers. And those peripheral terminals are not associated with any specific structure or mechanoreceptor or anything else out in the skin. They're just kind of free and wandering out and innervating whatever they can, like those tree branches we saw early on in this lecture series. Now there are, again, this is a figure that is representative of information that you learned earlier on about the different mechanoreceptors. And here are some examples of, on this graphic, what a C fiber and an A delta fiber look like. Those C fibers lack myelin. Their peripheral branches make this kind of complex branching pattern that crosses from the dermis into the epidermis and can actually get quite close to the outer layer of the epidermal skin structures. And A delta fibers do so similarly as well, except that they have a little thin layer of myelin. Now this is a histologic slide that shows an example of what those free nerve endings look like out in the real skin, out in the real epidermis. So you can see how close they get to that outer layer of the epidermis. And in fact, these free nerve endings, they go all over your skin. But what is important to note is that these little tiny branches, these complex branches, they all feed back into one main peripheral branch of the nerve itself. And so, therefore, what we can say are that free nerve endings, not only do they lack specialized receptor cells, they also lack encapsulations and they lack a specific mechanoreceptor. They are truly free to innervate the periphery according to the programming set up in neurodevelopment. But when it comes to free nerve endings, and more importantly, the peripheral nerve itself, from which those free nerve endings are branched off of, they can be characterized further by two very important molecular markers. And those are categorizing them as either peptidergic or non-peptidergic nociceptors. The peptidergic nociceptors are so named because they express and release neuropeptides. Neuropeptides are short sequences of amino acids, that's why they're called peptides rather than proteins, that are expressed by neurons, and in this case, by peripheral nerves. Now, there are two very important neuropeptides to draw your attention to, and they are substance P and CGRP. You'll learn more about CGRP later on when you learn about calcitonin in endocrinology. Now, most of our visceral afferents, so most of the nociceptors that innervate the visceral organs, and about half of those that innervate the skin, are themselves peptidergic. So what does that mean? Now, I mentioned earlier on that neuropeptides can be released as a result of afferent stimulation, particularly those when stimulated by those TRP receptors. And those TRP receptors activating a peripheral afferent can therefore also and subsequently induce the release of neuropeptides back from those free nerve endings back out into the tissue that they innervate. Meaning neuropeptides act kind of like a neurosignal. If we were talking from one neuron to another neuron, it would be a neurotransmitter. But in this case, neuropeptides can go out into the tissue and talk to immune cells. They can do all kinds of signaling cascades. And this is important for things in systems that involve chronic inflammation as well as visceral pain states. And it does so because chronic inflammation and ongoing stimulation of nociceptors that innervate the viscera upregulate as well as induce the release of further neuropeptides, which just kind of kicks off a signaling chain. Now, the other type of molecular marker that is important are basically all the other afferents. If they're not peptidergic, then they are non-peptidergic. That means they don't express neuropeptides. They don't release CGRP or SP. Now, there are very few, it's kind of rare, but there are some, but there are very few nociceptors that innervate the viscera that are non-peptidergic. And then if about half of the cutaneous free nerve endings are peptidergic, then the other half is about non-peptidergic. So these non-peptidergic neurons don't particularly contribute to visceral pain states, but they're more involved in somatic chronic pain states, things like diabetic neuropathy and some of the other kind of ongoing chronic pain that affects the cutaneous body surface tissues. Now, I mentioned that if a neuropeptide is released by the nociceptor that is expressing it, if it is signaling from one neuron to another neuron, then it's acting more like a neurotransmitter. And if it's signaling from the neuron out into another tissue or another immune cell type or some other cell signaling cascade, then it wouldn't be a neurotransmitter. Neurotransmitters are one neuron talking to another neuron. Now, instead of thinking about the free nerve ending, so the nerve ending that is the very terminal aspect of an afferent nociceptor out in the periphery, let's go upstream, closer to the central nervous system, and think about the dorsal horn. So this will be the central terminal of a nociceptor. And what we're looking at here, and I think it's easier to look at this graphic at the top first, this is that hypothetical ending of the nociceptor. So this in the yellow is the nociceptor itself. This is the primary afferent neuron. It is a C fiber. It synapses with the secondary sensory neuron in this signaling cascade in the dorsal horn, which is shown here. And you can see the very end terminal on the presynaptic side, it contains a bunch of stuff. It does contain your classic neurotransmitter, glutamate, which is denoted by these small little clear vesicles. But then you also have these large, dense kind of bubbles full of little dots, and those are the neuropeptides. In this case, it's substance P. We can tell because of the marker that was used in the staining down below. So what happens upon the action potential reaching this terminal end of the afferent, we have an action potential comes down. This is our synapse. You're going to have a release of neurotransmitter and neuropeptides into the synaptic cleft, and then it can go on and it can signal onto the postsynaptic side here. If it is a C fiber, in this case, C fibers can be peptidergic or non-peptidergic. They can release both excitatory amino acids, in this case it's glutamate, and neuropeptides at the same time to induce signaling. If this was an A delta fiber, it wouldn't be peptidergic or non-peptidergic. Those are specific to C fibers, okay? A delta fibers release only excitatory amino acids, in this case glutamate. So if you have a question and it's asking about what nerve fiber type is this most likely to be, and it releases substance P or CGRP as a signaling transmitter, then it's going to be a C fiber. You know that for sure. Now let's look down here at these electron scanning images. I think these are really neat. This is essentially the real life version of what the schematic up here at the top is showing you. But here you can see these big, beautiful vesicles that are containing all of our neurotransmitter, and they're labeled over here on the side so you can see it a little bit better. But I like this one because you can really see the synaptic cleft really well. But on this one, you can see these dots. These are the glutamate or the excitatory neurotransmitter, and then you have these bubbles with what looks like dots on the inside. Those are all of your neuropeptides that contain substance P in it. So that concludes this section of the video. Check back for the next part, which we will go into pain modulating mechanisms.